All right, excellent. So we have a really cool session on you coming up next about cellular therapies, but there's nothing cooler than hearing from our patients. So Sally Dale, thank you again very much for sharing your story with us. Thank you. Sorry, let me get our notes here. So our first speaker in this session is Qing Zhang, who's going to speak to us about the preclinical development of novel immunotherapy targets and cellular therapies. There you are.
All right, thank you. So, Sally, what a beautiful story, inspirational. So that actually reminds us why we're here. So also, thanks, organizer, for the invitation. Thanks, Phil, for moderating. So today I'm going to talk about some of the preclinical development novel immune therapy and targets in cellular therapy. So as you know, that the approval of the immune therapy, Busulfan, as well as TKIs, are largely based on the basic research done in the lab, and then that leads to the revolutionary therapy as well as well-winning prize. So in this case, I want to review latest development on that front in terms of novel immune therapy target beside CTLA-4 and beside PD-1. So the learning objective today, I'm going to review some of the novel new potential immune therapy targets, and then I'm going to talk about some of those development in terms of how we can actually target those novel immune therapy targets. So before we do that, I just want to give an overview. This is a nice review published by David Brown and Abhishek Chakraborty, who is here. In this case, this is a nice overview, simplified version about the tumor cells, the immune cells interactions. On the top, you can see the PD-1 and CTLA-4. But on the other hand, there are also other immune modulatory molecules, including TIM3, TIGIT, and LIKE3, and many others that could be potentially play a role. So how could other molecules behave in terms of preclinical setting? Does that actually warrant the preclinical as well as the clinical developments of targeting strategies, especially in kidney cancer? That's what we're trying to address. So in today's talk, I'm going to go through some of those molecule targeting and actually evidence from the laboratory setting. So for example, the first one is TIM3. So in this case, this study published a few years back actually took the peripheral blood cases from either normal patients as well as those kidney cancer patients. Also actually isolates those tumor-infiltrated lymphocytes from those kidney cancer patients. So when they examine the TIM3 expression, as you can see, the CD8 positive tumor-infiltrated lymphocyte was actually showing upregulation of TIM3 in those tumor cases, suggesting that could be potentially interesting in showing these dysfunctional molecules in the TIL in renal cancer. So in terms of therapeutic targeting in these studies, they use TIM3 blocking antibodies. In this case, when they examine the interferon gamma, the active version of those immune stimulatory molecules, either in CD4 positive T cell setting or CD8 positive T cell setting, there's increased interferon gamma positive T cells, suggesting that might be potentially therapeutically targetable or beneficial. Also, LIKE3 in this case, using the same role in terms of tumor and immune cell interactions, LIKE3 act as immune inhibitor molecules, which actually decrease T cell proliferation and inhibit those cytokine productions. So how about the LIKE3 blocking agents? In this case, this paper published showing that either using PD-1 blocking agents or PD-1 plus LIKE3, they examine the interferon gamma in CD8 positive T cells. When they examine the percentage of those interferon gamma CD8 positive T cell population, so you can see that they claim that there's benefit in terms of blocking agents. But ladies and gentlemen, I want to draw your attention. When you look at 10.3% versus 11.7%, I don't think that's actually significant, despite the paper claims to be so. So this actually, I want to refer back to my old boss, Bill Kading, who I trained with, that if you're actually going to make a career out of those kind of differences, I don't know how patient would potentially respond or benefit for that kind of therapy. So I do put a question mark in terms of how they could be potentially beneficial. Another one would be anti-VISTA. As you can see, VISTA not only expressed in the immune cells, but also promiscuity is also expressed in tumor cells. So in terms of VISTA blocking antibody, this study done actually in wrinkle cells when they use either PD-1, VISTA, or a combination. So you can see that when you combine double agent versus single agents. You don't see distinct differences, suggesting at least in this RINCA imperfect models, there's no therapeutic beneficial effects in this combination. Excuse me. The next one I want to move on is the new agent, which is in actually phase 1 clinical trials done by the Lawrence Fong at UCSF, moving to the Seattle at this time. So in terms of adenosine receptor pathways, as we may know, excuse me, it's majorly regulated by hypoxia signaling pathways. We know that hypoxia signaling is also one of our major focus in the lab. Hypoxia pathways can activate CD73 pathways. CD73 will bind to adenosine receptor pathways, will promote those immune inhibitor molecule formation, second AMP signaling pathways, which will trigger those inhibitor immune cytokine formation, therefore inhibit immune response. So in this case, these clinical trials, the phase 1 clinical trials, using the this A2A receptor antagonist. Thank you. I'm too nervous for that. So they use this A2A receptor antagonist, several adenoids, in this case, either in combination with the PD-1 blocking agent or single agent alone. On the right, you can see the waterfall plots. There's actually robust responsiveness of those patients, especially in the refractory renal cancer cell setting. So you have to argue that in this case, the single agent A2A antagonist may have to be combined with HIF antagonists in combination in order to boost this immune response. But at least this single agent suggesting that there's a rationale of this legitimacy targeting this A2A antagonist pathways in renal cancer. So in addition, we also want to talk about IL-1 beta. Some of you may actually see recent Nature paper actually using IL-1 beta blocking agent in pancreatic cancer setting. There's therapeutic efficacy. IL-1 beta is actually also hyperactivated especially in hypoxia signaling pathways. So in this case, it makes sense because those hypoxia signaling pathways can promote the immune suppressive signaling pathways in cancer. So we know that renal cancer is very pseudohypoxia due to the VHL loss. IL-1 beta, in this case, activates those reverse signaling pathways, including CRAB, FOS, as well as NF-kappa B signaling pathways. And in this case, in renal cancer setting, in this study, they use actually PD-1 blocking agent alone or IL-1 beta anti-IL-1 beta antibody, as well as in combination. Here also in the wrinkle cell model, they said that with combination of PD-1 and IL-1 beta, there's decreased tumor growth over time, suggesting that actually not only in renal cancer, but also in various other cancers, anti-IL-1 beta could be potentially therapeutic efficacious. So TPK1, which my lab published a couple years ago in Cancer Discovery, suggesting that actually autocrine signaling pathway, TPK1, can be therapeutic beneficial. It's also synthetically cited targets with VHL loss driven by oncogenic signaling triggered by TPK1. But in this paper, latest paper published by Robert Manguso's lab at MGH, suggesting that TPK1, when they use this patient-derived models, and then they actually look at those patients' relevant system, either using PD-1 alone, TPK1 inhibitor, or in combination, when they look at those immune response as well as therapeutic efficacy in various cancer, including kidney cancer, you can appreciate this waterfall plots showing that with combination therapy, TPK1 inhibitors can actually boost the therapeutic efficacy in various cancers, including renal cell carcinoma, suggesting this may be another molecules we should actually pay attention in the combination therapy setting. So last but not least, I want to talk about the endogenous retroviruses. This has been a hot topic recent years. So in this case, Ken Rasmus' lab published a few years back suggesting in those patients with partial response to immunotherapy, they're showing this high ERV expression. Also another study by the Vincent's lab at UNC also suggested the same thing, although in different ERV molecules, those partial response showing this higher upregulation of the ERV. And in those patients, also actually identify a potential antigen that is actually in CCRCC tumors that is upregulated. In these clinical trials from the NIH, when they actually look at non-myelolytic ablative hematopoietic stem cell transplantation patients in renal cell carcinoma, they actually observe a couple patients with amazing tumor regression. From those two patients, they isolate antigens that is common between those two patients. And then when they use those 10 amino acid peptide isolates from those antigens and look at interferon gamma response, and then they say there's an amazing response of those actually 10 amino acids ERA peptide that could be potentially use a potential antigen in CCRCC. So in summary, I want to say that immunotherapy in combination of therapy will be the key. There may be a lot of potential novel immunotherapy that we can pursue. Also, I think that combination of therapy will be the key to combat either primary cancer or potential resistance. Thank you so much for your attention.
Excellent talk. Thank you very much, Quing. We'll have Mike Hurwitz from Yale, who's going to talk to us about the CAR T landscape. Mike.
All right. Slightly blind, but that's okay. Okay, so here are the learning objectives. First off, thanks for inviting me to give this talk. Learning objectives are to hear about CAR T and kidney cancer. So there you go. And the take-home message, which is, what does this mean for my practice on Monday, is nothing, because none of these things are approved. So that's also kind of simple. So before I actually talk about the trials, I would like to talk a little bit about some basics of CAR T. Even though I think a lot of people know it, I would imagine some people don't totally know it. So we'll start with the T-cell receptor, which is how the T cell actually recognizes things that it wants to kill. And the important things to know are that the T cell receptor only recognizes peptide antigens, and they have to be presented by something called MHC or HLA. And the second thing that's really important to know is that you need a co-stimulatory signal. So if the T cell sees something, it won't really do anything for the most part unless there's another signal there telling it to do something. And finally, it's important to know that the signaling is through these blue things, the zeta chains. Now, antibodies are totally different. They can recognize any antigen, lipid, sugar, protein, and they don't use MHC. And I think one thing that people may not be familiar with is that in general, they have much, much stronger interactions with their antigens than the T cell receptor does with its MHC antigen combination, like 1,000-fold or 10,000-fold. So someone had the right idea of making a T cell function better by taking the business end of a antibody and sticking it on the signaling end of a T cell receptor. And that was the first generation CAR. And it didn't work, or it barely worked, until someone had the bright idea to say, well, we know you need co-stimulation, so why don't we put a co-stimulatory domain on it? And these are the CARs that we basically use these days. There was a third generation where they put in two different co-stimulatory domains, CD28 and 41BB, but it didn't really do a whole lot more. And then there are things called fourth generations in which they add in other genes. I think the most familiarly people refer to these as armored CARs. Okay, and then when you actually do it, it's a pretty big process. For the most part, we use T cells from the actual recipient because you have to have some sort of matching, or else the T cells will recognize your body as foreign, and the body will recognize your T cells as foreign, and they will kill each other. So this process takes about 3 to 4 weeks because you have to isolate T cells from a patient, activate them, transduce them with the actual CAR, and then expand them. And then at the same time, or near the end of that, you actually lymphodeplete the patient for a number of reasons that helps the T cells go in and stay functional. All right, without going into great depth, there are a bunch of unique CAR T toxicities or engineered cell toxicities. I think people have heard of CRS, cytokine release syndrome. And basically, these are all sort of the same sort of thing. ICANS or CRESS is CRS in the brain, and then a particularly virulent form of this is HLH and MAS. And there's standard treatment protocols, et cetera. All right, so here's actually what's ongoing in CAR T cells and in CAR T trials. And I just pulled this off of many searches into clinicaltrials.gov. The ones I'm going to concentrate on today are the CA9 and the CD70. Those are the only ones for which we have results. And I'll start with the anti-CA9 one. There are a number of ongoing trials, but this is an old one, and it's really more of a cautionary tale. So I think we all know CA9 is a good target for kidney cancer. And the reason it's good, of course, that it's highly expressed in kidney cancers, but it's also expressed at lower levels in the biliary epithelium. And the result of this is that when they did this study, the first 8 subjects, 4 of them had dose-limiting liver toxicity. And so what they had to do, actually, is pre-treat patients with a monoclonal antibody that blocked CA9, and the result was there were no objective responses. This is a very old trial. There were a lot of other issues with the actual CAR, et cetera. But this is a very important issue, which is what we call like the on-target, off-tumor effect. There are various names for it. And you got to pick your target incredibly well. CD70 is probably a better target for a lot of reasons. Without going into detail, if you look at part C of this figure from the paper by Panowski, you'll see that anything that isn't white on that part C is expression at some level of CD70. So clear cell has lots and lots of it, and some of the other RCCs do as well. So I'm going to talk about 2 trials. The first is CTX130. This is the COBALT trial that Monty Paul presented last year at SITC. And this is actually different from what I presented. This is an allogeneic T cell. So how do you make an allogeneic T cell actually work? Well, what they've done is using CRISPR, they have knocked out beta-2 microglobulin, which means that it doesn't make MHC class I, and they've blocked their own T cell receptor. They've inserted something into that. So it won't recognize the recipient as foreign, and the recipient, for the most part, won't recognize it as foreign. And the advantages are you don't have to wait 4 weeks to get this into patients. The cells that you get are from healthy donors, so they're an intact immune system, and you could make tons of it. The efficacy was not massive. I would say it was moderate. There was 1 response. There was about 70% stable disease. And if you look at we don't have a formal duration of response, but it wasn't massive for most of them. Safe, though, there was CRS, about 50%, but it was only grade 1 or 2. And basically, people tolerated it very well. All right, the second trial is the TRAVERSE trial that was presented by Summer Sroor at AACR this year, and in a way, a very similar trial. This is also an allogeneic, also anti-CD70, but there were some key differences. So what they did is they knocked out the T cell receptor so that they wouldn't recognize the recipient, and they knocked out CD52. And the reason they did that is they could use an anti-CD52 antibody, which is a lot like alemtuzumab, but it's theirs. They call it ALLO-647, and that's very, very immunosuppressive. And they do that to make sure that these T cells stay around. They also knocked out CD70 because although CD70 is highly expressed in kidney cancer cells, it also is expressed, upregulated actually, on active immune cells. And they were worried about fratricide. That is, while they're growing them up, the T cells killing each other. It was also pretty safe, although again there was a fair amount of CRS, only 1 grade 3, and only 2 in neurotoxicity. So again, it was a pretty well-tolerated drug. But what they saw was a much better response, frankly. So in all patients, there was about a 17% response rate. And in the ones that were expressing CD70, there was a 30% response rate with a disease control rate of 100%. And you can see it here in the waterfall plot. Everything on the right is CD70 positive, and it's a pretty impressive result. Again, we don't have an official duration of response, but it looks like it might be significant, certainly in at least 1 patient. And am I running late? I don't know my time. I'm good on time? Okay, so then I'll talk about the other 2 things that they found. So one is that they can show by this spider looking plot, that's actually presence of the cells at time. And what they found is that even 70 or 80 days out, if they use like a very strong conditioning regimen, these things stayed around a long time. And we know that durability of response has to do with persistence of the cells. And the last thing that was sort of interesting is what they found is that the CD70 had this important effect, which is that when you infuse the drug, the alloreactive host T cells upregulate CD70, and then they actually get killed, which allows the T cell response to go on longer. And then eventually, these anti-CD70 cells go away, and you can see at the very end, regular T cells are coming back up. That's the purple line. So to conclude, this is exciting. There are definitely some responses. We're not there yet. But there are these key challenges for solid tumors in general that are going to make it a lot harder than it would be for liquid tumors. It's hard to get these T cells to persist. solid tumors, of course, can lose any antigen, and so that's going to be hard to really kill off a tumor that way. The microenvironment is very bad. It's hard to infiltrate. So what I would say is that it's exciting, and these are interesting. We're not there yet, but just like the end of every Hollywood blockbuster, when like the bad guy is dying and is like falling into lava or something, he always like, as his hand is sticking out, he goes like, this is just the beginning. And I think this is kind of just the beginning, because there are many, many different technologies we can use to make these T cells much more effective. Thanks.
Great, and to wrap up our session, good friend and colleague from the University of Pennsylvania, Vivek Narayan.
Thanks, Phil, and the organizers for the opportunity to present on novel immune therapies in early phase clinical development for kidney cancer. I think really building on what Dr. Zhang presented in the first talk and focusing on how we can actually do clinical translation of some of the targets he highlighted. So I thought as objectives, I would first start with thinking about how we can adapt a kidney cancer immunity cycle to potentially inform novel immune therapy development in RCC. And then second, rather than provide an exhaustive review of all the immune therapies under active clinical development, try to highlight some key representative therapies under development that may seek to target various aspects of this adapted cancer immunity cycle. So first, just a brief background slide. It's clear that we have significant unmet need in therapeutic management of advanced RCC. Although we have a number of recent approvals, these are largely associated with only incremental benefit, and we still have a lack of options beyond standard VEGF receptor TKI and immune checkpoint inhibitor-based approaches. And for the vast majority of patients who receive a combination immune checkpoint inhibitor-based frontline approach, primary or acquired resistance after an initial therapeutic response is unfortunately invariable. Yet we know that kidney cancer is an immune-responsive tumor. We've known about the historical efficacy of high-dose IL-2 in this disease, and more recently with durable clinical responses to combined immune checkpoint inhibition, and certainly stories like Sally just shared about really durable responses to anti-PD-1 therapy. But we also know there's a lot of disease-specific challenges that may limit cancer immunity in kidney cancer. So for one, there's relatively low or middling tumor mutational burden relative to some other malignancies. And the particular cancer-specific antigens that may drive an anti-tumor immune response still mostly remain unclear. Kidney cancer can be plagued by ineffective antigen presentation, and although there can be areas of high T-cell infiltration, this doesn't always appear to be associated with clinical response to checkpoint inhibition, and there can be considerable heterogeneity in advanced RCC, with immune-infiltrated versus angiogenic subtypes, as well as variable immune cell phenotypes and immune cell metabolic functions. And so as I thought about preparing this talk on novel immune therapy development, I thought a useful framework would be this adapted kidney cancer immunity cycle proposed by David Braun and others a few years ago. When we think about the necessary steps for effective cancer immunity, moving from the release of cancer cell antigens and effective cancer antigen presentation, to priming and activation of immune cells, trafficking and infiltration into a tumor microenvironment, and ultimately cancer cell cytotoxicity, we can think about unique aspects of kidney cancer that may either promote or inhibit effective cancer immunity. So for example, the relative paucity of tertiary lymphoid structures in kidney cancer and how that may affect antigen presentation, factors within the tumor immune microenvironment that could limit the effective cancer infiltration of effector T cells, and certainly genomic abnormalities that could potentially be associated with a more favorable response to immune checkpoint inhibition or perhaps an unfavorable response to immunotherapies. So we think about this adapted cancer immunity cycle, we can think about now novel therapy development and how it may seek to exploit various aspects of this cycle. And I think the first place to start is beginning with novel immune checkpoint strategies and targets. So we know that one potential factor that can limit the efficacy of immune checkpoint inhibition is the presence of regulatory T cells in the immune microenvironment. And so building on that is this phase 2 study led by Dr. Atkins and others, seeking to evaluate botensilimab, which is a next generation anti-CTLA-4 antibody that has an enhanced FC portion with the idea that this may allow for more selective or high affinity binding to FC gamma receptors. So the clinical hypothesis here is that this may optimize engagement with APCs, thereby better promoting antigen-specific T cell responses, and also preferentially depleting regulatory T cells in the tumor immune microenvironment, thereby enhancing the effector to regulatory T cell ratio. This would be combined with a novel anti-PD-1 antibody, and in this randomized phase 2 study of 120 patients would be compared against the control arm of ipi-nivo with the primary endpoint of objective response. Staying on the same topic of novel PD-1 CTLA-4 strategies, we saw within this past year results from a phase 1 first-in-human study of a kidney cancer cohort of 46 patients with MEDI5752, a novel bispecific antibody, where at least in this initial experience, there was some promising and durable anti-tumor efficacy, particularly in the first-line treatment setting, with objective response rates approaching 60%. But this was also associated with relatively high rates of toxicity-related discontinuation, including hepatotoxicity, and so this compound's being evaluated in various combinations, including with VEGF receptor TKIs in the frontline setting. And then we, of course, as Dr. Zhang highlighted, we have a number of immune checkpoints beyond PD-1 and CTLA-4, including TIM-3, TIGIT, and LAG-3 inhibitors building on the melanoma experience with nivolumab and relatlimab, here in combination with zanzalintinib, XL092, in the frontline clear cell RCC setting. So coming back to the adapted cancer immunity cycle, what about building on the anti-CTLA-4, anti-PD-1 backbone and focusing on immune metabolic targeting? And Dr. Zhang talked about the importance of the CD73 adenosine pathway in immune metabolism and how adenosine and A2AR binding on dendritic cells, macrophages, T and NK cells can potentially inhibit anti-tumor immunity. He highlighted how the small molecule inhibitor ciforadenant and inhibition of A2AR with Larry Fong and others a few years ago has shown some efficacy in clear cell RCC, particularly in combination with immune checkpoint inhibition. So building on this initial data, Dr. Yonash and Dr. Beckerman, through the Kidney Cancer Research Consortium, will be conducting this phase 1/phase 2 clinical trial testing ciforadenant in combination with ipilimumab and nivolumab at standard dosing in the frontline clear cell RCC setting with primary endpoints of safety, tolerability, and depth of response. And so just looking back at the cancer immunity cycle, it's clear that although we have novel immune checkpoints and immune metabolic targeting strategies under active clinical development, there's really a number of things targeting various aspects of this cycle, like therapeutic vaccines to enhance priming and activation, modified cytokine therapies like IL-2, building on the BEMPEG experience, but with novel compounds, immune co-stimulation agonists targeting 4-1BB and others to enhance cancer cell cytotoxicity, and we just heard from Dr. Hurwitz about adoptive cellular therapy in kidney cancer with CAR T cells. So what's the take-home message? What does this mean for the future of care? I think with novel immune therapy development in kidney cancer, it appears that we're really building on the effective backbone of PD-1 CTLA-4 inhibition, but I completely agree with Dr. Zhang that it's really going to require multifaceted approaches, I think really informed by an RCC cancer immunity cycle to ultimately improve clinical outcomes. Thank you and look forward to questions.
Amazing presentations, gentlemen. We'll open up the floor if anybody has questions or comments. Well, I'll ask you then as we go right down. What are you each of you, what are you most excited about in this area in the next 2 years?
For me, I think that's the combination with A2AR antagonist with immune therapy and or actually HIF-2α antagonist in combination will be actually very promising and very exciting.
I'm actually most interested in the ERV story. I think that, we don't exactly understand why kidney cancers are as immunogenic as they are. Understanding more of the basics of that might lead to sort of much more sort of durable things that we won't have to use as many toxic agents for.
I'll give a different answer and maybe share my bias, but I'm really fascinated about the potential for adoptive cellular therapy. I think, sort of optimistically, I think we're on the cusp in solid tumors, whether it's kidney cancer or other diseases, and the ability of a modular construct to really overcome some of the challenges that people have described. I think CARs have that unique ability.
That was supposed to be my answer.
I have a question about CAR T-cell in kidney cancer. Don't you think it's a little early to jump into this therapy, which is quite toxic, when we don't have a targetable antigen so far? I mean, CD70 has been used as with an antibody which doesn't work. CA9 so far is not targetable for treatment at least. So why don't we jump so fast in CAR T-cell outside of doing clinical trials?
So, of course, I have to say no. But sure, it's toxic up front, but frankly, it's not that toxic. Frankly, if I had my choice to get a durable response out of it, I'm not saying that we have durable responses yet, but if I could do that or go on, a TKI for the next X number of years, I think I would absolutely choose a little bit of toxicity up front. People aren't dying on these trials. They're going through a lot in the first week or two, and maybe the first 3 weeks, and then after that, it's comparatively benign. And also, these are trials. We're almost always doing these on people who've already been through all these things anyway. So no, I think it's reasonable to try things like CD70, CA9. Yes, I don't think that should be a main target.
I think your point's very well taken, that selection of antigens in a solid tumor malignancy like RCC is going to be different than how we think about it in hematologic malignancies. What is the cell surface expression? What is the density and specificity of the antigen? I mean, I think all of these things are fundamentally going to be different than we see in heme malignancies, and so there's still a lot to figure out on what really makes an optimal antigen for cell therapy in solid tumors. But I think the toxicity we'll figure out.
Yeah, a kind of a broad question here around immunotherapy development, because it seems like the clinical development is still following along the same lines as our drug development. We're fairly empiric. Take, come standard of care and add X to it. And I'm just curious, in the absence of kind of validated biomarkers, are adaptive approaches a better way to go here in terms of thinking about, using response to design where you add or switch or intensify therapy, or do you think that we'll make progress just with the more kind of standard empiric first-line, second-line approaches?
I'm happy to start. I think that's a great comment, Dan. It sort of builds on the patient story we hear. I mean, there's certainly people who have beautiful, durable responses with the conventional therapies we have right now. And so is there a sort of clinical biomarker or scientific biomarker that we use to de-intensify therapy, or do we escalate therapy based on ctDNA or some imaging biomarker or clinical biomarker? Absolutely. At the same time, though, novel therapy development, I mean, we know that the vast majority of patients are going to get acquired resistance to these. And so what do we need to target mechanistically at other aspects of that immunity cycle to help those patients? I mean, I think it's really going to be a combination of both.
So I just want to add on top of that, carpet bombing, like immune therapy, definitely actually works, but only has its limitations. But at the end of the day, sometimes we need to see precise stratification of patients. So for example, the clinical trials, phase 1 in the refractory kidney cancer setting by Lawrence Fong studies, they actually chose those adenosine signature high patients treated with A2AR antagonists on top of that with the immune therapy. That proved to be efficacious. So I think that precision oncology plus those immune therapy will be important based on the stratification of patients.
Great, thanks.
Yeah, this was a wonderful session, really educational, informative for me. My question is about the target. We really seem to have moved on to CD70 as the next most likely candidate. But are there other, sort of potential targeted candidate antigens in the works that you think might be potentially better?
So I don't know. The answer is that I've been wondering the same thing, and I haven't seen a whole lot. I personally don't work on target development. I'm sure there are other targets out there, specifically in kidney cancer, but I don't know of any great ones. What do you know the other ones?
Yeah, I think that right now there's a lot of emerging development with the bispecific antibodies or actually hybrid cytokines, which they combine IL-2 with either CD70 or actually PD-1. So that would be actually something we should look into, because those take advantage of those bispecific or actually hybrid molecules, bring those tumor cells and T cells together, take advantage of targeting both and brought into then into proximity, so it gets very efficient cadence. We actually have some preliminary data in the lab on that front, but actually proves to be very efficacious. Thanks.
Maybe the only thing I'll add, Ben, is that it's, although CD70 by itself may not be a great target, CA9 by itself is maybe not the ideal target. There's different ways of combining or thinking about different cancer antigens, so logic gating, other things that can be done to improve specificity. So I think a lot of different ways to think about using different targets together potentially.
Yeah, I guess I would say that also that, right, so this idea of the logic gating, there's also tuning that you can do, so they can do something where you'll take a target that may not be as specific. The CAR T will recognize that target, and instead of that turning on the T cell, that'll go to a transcriptional program that'll turn on the very specific receptor, and then when that very specific receptor, CAR goes up to the surface, then it will kill off. And you can and when you do that, you can use targets that may not be perfect on their own, but together they're very, very specific and sensitive.
Thanks, and we'll do question, and then we'll take a break.
Thanks. One general question, the concept of bispecifics is one that has really taken off in other cancers, and in many ways with either radiopharmaceuticals, chemotherapy, and other cancers are just bispecifics targeting immune cells. Do you think, are you optimistic for, we didn't see a lot of that, in these talks about bispecific therapies, and these are off the shelf and often can be easier to give compared to CAR T. Do you see a future in RCC? It's more of a general question. And then just quickly, one specific question on ciforadenant. Do you think that there's monotherapy activity from the waterfall plots you showed? So I wondered, what setting do you think A2A antagonism would be best used in combination with PD-1 or in resistant as monotherapy potentially?
So take down the A2A antagonist with the study you mentioned. I think that that actually was done in a refractory setting, given the fact probably because those patients display very high signature for the hypoxia. So that's probably explained why those patients have high, the signature for adenosine. So that's how those patients could be responsive. But on the other hand, as I mentioned briefly, they only block part of those hypoxia signaling adenosine pathways. On the other hand, they actually didn't cripple the HIF signaling per se, because HIF signaling can also induce those immune response suppressive pathways. So I think it has to be combined with HIF targeting strategy or actually combined with immune therapy.
With the bispecifics, I sort of intentionally left that out just because I don't have a lot of firsthand experience with it, but I completely agree. I think it's an exciting avenue and certainly has shown some promise in other solid tumors. It sort of falls in that same category of T-cell redirection. I know that it's active. I mean, there's the PD-1 by CTLA-4 type bispecifics, but I think probably more what you're referring to is like the cancer antigen by CD3 bispecifics that are under development. I mean, I think Xencor is here and they have a product under development. So I know that that's an active space.
Yeah, I would just add that I think that they may take over from these basic CAR Ts, but I think that, again, what you can do with a CAR T, which is a little bit different, are these logic gating and the more complicated things. You can make a much more sort of complicated, tailored drug, which I think may be really the future, because I think BiTEs will do a lot of the same thing.
Great, thanks.
Martin Voss, New York. Great presentations, thanks. I have another CAR T-cell question. So for patient selection or the appropriate clinical space to develop these strategies, I guess two questions. For one, we talk a lot about tumor penetrance and the difficulties in solid tumor development. Do you think we should preferentially enroll patients with low volume of disease or specific organ sites? Has that been looked at in these ongoing efforts? And then secondly, another question that comes up often as we consider patients for clinical trial options later line, high-grade toxicity, so prior checkpoint inhibition. By mechanism, should these patients be okay to go on these studies, since we're now using very different approaches?
So to the first question, that was a major point of discussion, when we were doing the COBALT trial. Should we be looking at smaller volume? And I think theoretically, maybe yes, but I'm not sure how that eventually leads to the development of a drug if you have to limit it by those things. So, and we also, part of the idea behind, a CAR T is that, sure, you're going to get some killing, but the idea is that you'll get immunologic spreading also, that hopefully you will get a larger immune response by doing that. So, so even though, yeah, that's a major issue, I don't know the answer, obviously, but my bias is no, you shouldn't do that. You need a drug that's going to work. And then what was the second question? Sorry.
I think about the different metastatic sites and how that may influence thinking about CAR T cells. Is that right, Martin?
The second question was about toxicity to prior therapy, high-grade toxicity to checkpoint inhibition. Is that patient still a good candidate for CAR T?
Right, and the mechanisms are really quite different. So I don't worry so much about that. It's really almost like a cytokine toxicity versus, your standard IO type toxicity, which is probably from, again, activation of other parts of the immune system or you know what I mean? So I don't worry about that so much, and I don't think we've seen that. We've also, when using...
Do the trials allow for it? Do the trials allow these patients to...
Oh yeah, absolutely. All these trials actually kind of demand it, that you've been on IO already and TKIs.
Nizar, last question for the group.
Great session again. You spoke about CAR T bispecific. What about CAR NK? At MD Anderson, obviously, the success has been in hematological malignancies, the paper from Katie Rizvani's group at MD Anderson and New England, I think a couple of years ago. Do you believe, CAR NK would be a viable option, similar to CAR T, maybe less toxic? Obviously, again, it's the antigen, like you already articulated, all three of you, about the antigen and the importance of identifying an antigen. But if you have that perfect antigen, do you think our CAR NK would be a better strategy?
Yeah, Dr. Rizvani's data are really impressive in CLL. So CAR NKs are an excellent idea. There are now CAR macrophages. And of course, there are CAR T cells using gamma deltas versus alpha betas, and they all have somewhat different phenotypes. All of them can kill. I don't know what's going to be the winner, but I think they all probably they may all have uses in different diseases somewhat differently. I don't know if you have a different thought.
Not much to add. I think there's a lot of theoretical advantages to CAR NK in terms of potentially favorable toxicity profile and multiple mechanisms of cytotoxicity. I think in solid tumors, it's still very early, but I think certainly something that it needs to be explored and which should be interesting.
Good. I want to thank the audience for their attention and specifically for not asking me any questions during this session. That was great. We're going to take 10 minutes and come back with wood fire. It's going to be awesome. So 10 minutes, get your snacks, come back.